So this mini lecture on T cell development and the thymus and autoimmune risk is a conceptually important part of the unit because it explains how the body trains T cells. And if we understand this selection process, then we're much better prepared to understand both normal immune defense and kind of the major pathway into autoimmune disease. So kind of the premise of today, we'll get through discussion of thymus selection and then how that process works to train T cells to recognize threats. So let's start with T cells. T cells, they develop in the thymus, hence T cell. The thymus sits in the mediastinum of the chest, so right there in the center, and these cells are the major players in cell-mediated immunity. So unlike antibodies, which are floating around in body fluids, the T cell receptors stay attached to the cell surface. And that distinction is important because the T cells are designed to recognize presented antigen directly through the cell surface receptors. So the body generates an enormous variety of these receptors by shuffling gene segments and modifying them in complex ways, but the result is a tremendous range of possible receptor combinations. Now, the body cannot keep all of these at the same time, so some receptors don't work at all. Others bind too strongly, and if they bind too strongly to our own proteins, that is a dangerous recipe for autoimmune dysfunction. So the thymus becomes the training ground for your T cells. It is a way for the T cells to then test the receptors against self components, and that happens in the thymus. So essentially what we're looking at is T cells are trying to observe whether or not they're going to recognize self-antigen. And what we're doing is determining whether or not these T cells bind effectively. And if they do not bind effectively, they're not useful at all. If they bind too strongly, then they're a threat because that means they're going to bind strongly and identify us as a problem. And that search and destroy for self is autoimmune disorder. So there's a sweet spot, and that is weak recognition or weak binding. Weak binding and weak recognition means that they can interact appropriately without launching destructive attacks on yourself. And this is why the thymus is so important in preventing autoimmune disease. So imagine you're a T cell sitting inside the thymus and there's not a lot of infection going on. Well, every cell in your body is regulated. And since the T cell is not needed, after a few days, the T cell dies. In the thymus, the T cell is testing the receptors against proteins that naturally exist in the thymus gland, our own proteins. So they're testing the receptors by presenting self-antigen. So as I mentioned, the T cell can't if the T cell can't bind anything, then it dies after a few days. Now, the assumption is the receptor didn't work, so the body gets rid of it. However, if the T cell finds self-antigen that's presented and it binds self-antigen strongly, then this T cell would be destroyed because it is binding strongly to our own cells. That's a recipe for autoimmune disease. So if you have a receptor that binds really well to stuff in the thymus, that'd be a bad receptor to keep around because it is going to find a way to bind or call out self, and we're going to develop an immune response to your own cells. T cells that bind our own proteins really well, not very good. We need to get rid of those, and that happens via apoptosis. So we don't want T cells binding to self-antigen, indicating self is foreign. That would not be a good recipe for success. So receptors that we want to keep are the ones that bind weakly. Or not as strongly. So if we keep the strong binders, that's a risk for autoimmune disease. If we keep the ones that don't bind anything, that's not a recipe for success because they're not going to find anything. We don't want to keep them around. And so some people are not as good at getting rid of strong binders. That predisposes them to disease. Consider somebody with multiple sclerosis or lupus or Crohn's disease, which are all autoimmune diseases. If we have too many T cells binding strongly with self, autoimmune dysfunction results. So let's talk about selection of T cells. And this slide here gives us some of the, call it the vocabulary of T cell selection: death by neglect, positive selection, and negative selection. So positive selection means a weakly binding T cell is kept for further development. Death by neglect, this refers to cells that essentially fail the test because they don't bind and they're not useful to keep around. Negative selection is when the body deliberately eliminates a strongly binding T cell because it is too reactive, and too reactive against self-antigen is a recipe for autoimmune disease. So the phrase I want you to keep in mind is that weakly binding is best, or weak binding is best. Not zero binding, not strong binding, weakly binding. Now, the military academy analogy works pretty well here. If you think of this, the thymus as the training camp, the entrance exam asks whether the cell can recognize the appropriate uniform. In other words, can the cell recognize self-MHC, or self-major histocompatibility complex? The ethics board is then going to ask whether the cell can do their job without attacking civilians, meaning healthy self-antigens. If the cell fails either side of that process, it can't recognize our own uniform, or it attacks civilians, meaning it can't recognize self-antigen, so it doesn't have any binding, or it binds too strong and starts attacking self, then that cell does not move forward in the selection process. If the body fails at negative selection and it ends up keeping strong binders, that becomes a mechanism for autoimmune disease. In other words, the wrong graduates are allowed to leave the academy, and those wrong graduates attack self. That's not what we want to see happen. So if we fail to get rid of the strongly binding T cell, that's a failure and a mechanism for autoimmune disease. We'll keep in mind, kind of as a summary, that the thymus is like the military academy. The entrance exam talking about positive selection. So in other words, prove you can recognize this nation's uniform. Prove that you can recognize self-MHC. The ethics board is going to step in if you prove that you won't shoot civilians or you won't attack self, then we can keep you around. And then we start to have our branch assignments. And this is something that we'll get into a little bit later. The branch assignments now start talking about a distinction, whether you're going to be in the infantry, your CD8 cells, or whether you're going to be command and control, like CD4 cells. So graduates are then going to patrol the home base. They're going to hang out in the lymph nodes until they're given a mission dossier, an antigen. They're also going to be given rules of engagement, right? Co-stimulation, how do we activate? Where do we need to go? What does this look like? So this is kind of going to be the foundation for our understanding of this T cell selection. So once these cells are properly selected, the T cell leaves the thymus. They're still considered naive because they haven't yet encountered their real target. These graduates from the academy have not yet encountered real combat. So they're going to travel through lymphatic channels and eventually into lymph nodes. And this is where they're going to wait for meaningful antigen presentation. So antigen presenting cells from the innate immune system, we know these as macrophages, neutrophils, other dendritic cells, and they're going to travel to the lymph nodes. Now they're going to present antigen to the T cells. If a naive T cell recognizes a foreign antigen strongly enough in the right context, it's going to become activated. And it says, hey, I've recognized something that shouldn't be here. So it's going to start proliferating. It's going to make copies of itself. And then those activated cells then leave the lymph node and start to carry out whatever effect or function we're talking about. So this process, of course, takes time. This is why adaptive immunity is not instantaneous. So a good practical example is someone with strep throat or similar exposure. A person encounters a pathogen today, but the full adaptive response still requires processing, presentation, activation, proliferation. So when you think of lymph nodes, think of them as the meeting points, the gathering site where the immune information gets exchanged and decisions are going to get made. Remember, T cells with receptors that don't bind and T cells with receptors that strongly bind, they're destroyed. So they're not playing a role in this process. Now, if we have a cell that has developed a receptor that's capable of working, we said this was a weakly binding receptor, but it hasn't been tested against the real pathogen, again, we call that the naive cell. So naive cells have gone through basic training in the thymus, but they passed the selection, they passed the ethics board, right? They were not destroyed due to no binding or too strongly binding to self. But these T cells still need to be combat tested. And so keep this in mind for the process. It does take a couple days once that infection is identified for those cells to proliferate. So if these cells have been through basic training and then undergo a response, they're going to start to proliferate. And this is where they're going to start recognizing and making copies so that they can then enter into the system and start to fight the infection. So this slide introduces the presentation step a little more directly. Antigen-presenting cells are going to digest pathogens into smaller peptide fragments. And those fragments are then loaded onto molecules called major histocompatibility complexes, or MHCs. And these are going to carry the peptides to the cell surface. A simple way to picture this is to imagine the MHC as a sign holder. It grabs the peptide or sign and it presents it in a way that a T cell can look at it, inspect it, observe it. And if the T cell receptor is not just looking at loose antigen floating around, it's looking at antigen being shown in a proper presentation format, this is where it's going to identify, recognize, bind or not bind, or too strongly bind. So this matters because T cells require context here. They're not meant to react to every free-floating molecule that they encounter. The T cells respond when antigen is displayed in the right platform on the MHC, or the MHC displays it. So, if you ask the question, well, how does a T cell recognize antigen? The answer is not just because antigen's there. The answer is because the antigen has been processed, it's been loaded, and it's been presented through major histocompatibility complex, or MHC. So the T cell receptor evaluates antigen only when it's displayed on the right platform. Now, the takeaway from this section or discussion of T cells is that T cells are not simply born useful. T cells have to be trained, they have to be tested, they have to be selected. Weak binders are kept, dangerous, strong binders are eliminated. And once a properly trained T cell meets real antigen in the lymph node, then it can expand into an effective response. So, how do antigen-presenting cells present antigen to T cells? Remember, a protein, it's like a holder, binds to a peptide and travels to the cell surface. The green MHC that you see on the slide, this is the antigen presenter called MHC. Now, MHC holds a peptide, shows it to the T cell to be recognized. T cells that do not respond to loose antigen alone, right? They're trying to inspect and identify whether or not any binding is going to happen. So this is all the process where we're trying to identify how do we move these T cells forward.